All right, so what we'd like to do is start our section on DNA. This is chapter 16. We're going to look at the molecular basis of inheritance, or really what we're going to look at in this chapter is DNA replication. So here is the molecule itself, and it's showing replication. We're going to go through the structure of the parts. So here is the reminder from test one, the nucleotides. There are 5 of them. There are pyrimidines, the longer word, simple structure. So there's a single ring. And that is going to include cytosine and thymine in DNA, and uracil in RNA. Then we have the purines, which is a double ring structure, adenine and guanine, and that is for both DNA and RNA. And then the sugars, deoxyribose, the pentose or 5-carbon sugar for DNA, and ribose is the pentose or 5-carbon sugar for RNA. So all of that is review. Okay, so let's talk about the new stuff. Let's talk a little bit about the history. So 1907, T.H. Morgan, working at Columbia University, actually coined the term genes. He was looking at early fruit flies. The reason he chose fruit flies is that they have 4 chromosomes, so easy to identify and manipulate. Humans, we have 46 chromosomes, 23 pair. And so anyway, so he came up with the idea of genes. And then when we get into the next test, we'll talk about genotypes and phenotypes. And genes that are expressed are phenotypes of the characteristics that appear. So we'll discuss that later. But first man early on to really work on this stuff. And then in 1928, transformation by Griffith. And what I want you to know about Griffith, this is a pretty famous study in the fact that he actually proved that information can be passed on, can be inherited. So we want to take a look at this experiment. Obviously, he worked with white mice. You can see them in the picture. But he also worked with bacteria strains, and for whatever reason, they don't mention it, but these are pneumonia. All right, and this pneumonia strains come in 2 forms. One was the living S, and one was the S strain and the R strain. And the S strain looked smooth under the microscope, and it looked smooth because it had a protein capsule around it. And then in the rough ones, they looked rough under the microscope because, again, they lacked this capsule. They didn't have this protein capsule. So what he actually did was pretty smart. He injected both different strains into mice, and what it found out is it was the living S strain that actually killed the mice from pneumonia, and the R strain would stay healthy. So he was wondering why. So it had to be that capsule. What he ended up doing was taking the strain of pneumonia that was killing mice, and he heated it up. And by doing that, he denatured the capsule, and it turned out that that could be injected into the mouse, and the mouse would live. So it was actually something about that capsule that was causing the deaths. And then where he got really smart, and where he is well known for, is what he did then is he took the living R strain and exposed it to not the living S strain, but the heat killed S strain. So these things are dead. They don't have the ability to kill anymore. But by exposing the R cells, the rough cells, to this killed S cell, they actually picked up information and then was able to actually, in the next generation, be able to kill the mouse. So these R cells are picking up genetic information and then being passed. So this was the first proof that genetic information can be passed on from one organism to another, one individual to another. All right, so he was using bacteriophages, which is phages are viruses that attack bacteria. And again, the virus has DNA or RNA, and it's most of them are covered with a protective capsule or coat. And geneticists have used these a very long time before experiments. So, you know, since the 20s. So pretty cool. All right. What I need you to understand is the history of what's going on here. And so once they figured out information can be passed, they assumed it was in the chromosomes. But remember, chromosomes are made out of two things. They're made out of DNA, and they're also made out of a histone protein. And so there was questions for years on what is the most likely candidate. Now, today we know it's the DNA, but back then we weren't sure. And matter of fact, if you look at kind of logically, the assumption was it was protein, and that was kind of the leading idea at the time. And the reason makes sense. If you think about it, DNA, the nucleotides, there are 5 of them. There's only 5 different types, 4 of those in DNA. Whereas in protein are made up of amino acids. And there are 20 different amino acids. So the assumption was that, you got 20 of them, more likelihood to show all the diversity that we see today. Avery, McCarty, and McLeod in 1944 wrote an article on the pneumococcal types, basically looking at Griffith's experiment, and they suggested that it's actually the DNA, not the protein. Now, they didn't really have any proof, but they were saying it could be very similar to what viruses were doing. So they weren't 100% sure, but they were suggesting it, and so this was really kind of the first push for this. I do want you to know Hershey and Chase, and these are Hershey and Chase here in the picture, and they were working here in California, and there in 1952 they came up with the idea that it was actually the DNA. So they were the ones to prove basically the paper. It took them 8 years to actually prove it. So what I want you to know is that they did this very famous study, and it was pretty smart. What they did is they made some of the stuff radioactive. And so they wanted to see where that ended up in the new individual. So what they did is they made things radioactive. Now, what I want you to know, and we haven't really gone over, is how these things separate. So proteins actually have, if you think of CHNOPS again, proteins have carbon, hydrogen, and oxygen, nitrogen, and sulfur. What they are missing is the phosphate. There's no P in protein. And then what happens if you look at a nucleic acid, it's made up of a pentose sugar, so it's CHNO. It's made up of a phosphate group, and it's made up of a nitrogenous base, so it has the N in there. So what's actually missing on there is the sulfur. So what they did is they made both of those radioactive, and then they let them breed, set them through, and what they found out is that it was the phosphorus that was ending up in the radioactive pellet, but it was the phosphorus that was actually ending up in the living matter. So they proved that it was DNA. All right. The one last thing I want you to know, and this was done in the 40s, but Chargas rule. What he realized is he did experiments where he put a bunch of, like he wanted to find out, you take a cat, not the full cat, but a cat cell or a dog cell or a person cell. and he put them in a kind of in a blender and mix it up. And then he would actually look at the amount of A, T, C, and G. And what he realized is adenine always equaled thymine and guanine always equals cytosine. So, or at least pretty close. So the assumption was that these somehow were bonding together. It's called Chagos rules. All right. So what I need you to know from this is there's a big, huge push to figure out the structure of DNA. And Linus Pauling had nothing to do with this. But I have him here because Linus Pauling was one of the very famous people that were working on it. So everybody knew that this would probably be a Nobel Prize. And so what happened was all these scientists started, all these labs were rushing to figure out what is the structure of DNA. Now, Linus Pauling has won 2 Nobel Prizes, one in chemistry on bonding, and then the other for the Nobel Peace Prize for his work on the Manhattan Project. And it wasn't really the work on the Manhattan Project, but the condemnation of the nuclear bomb and his fight for not using it. But anyway, so he is famous for the alpha helix structure. You know, we talked about alpha helixes and beta pleats and protein secondary structure. And so he actually at one point was working on DNA and assumed it was an alpha helix or actually a triple helix. That's what he was working on. So I'm just trying to show you that, you know, this is one of the bigger minds in science at the time. One of the only 2 people to win Nobel Prizes, 2 of them separately on their own. The other one is Madame Curie. And so, but anyway, so just to get you an idea that there was a huge race to determine the structure of DNA. And with that, we will stop this recording.